What is DNA replication?
DNA replication is the process by which a cell makes an exact copy of its DNA before it divides. This is essential for growth, repair, and reproduction. The process ensures that each new cell receives a complete set of genetic instructions, preserving the organism's traits and functions through generations.
Starting at the origin,
replication begins at specific locations called origins of replication. Proteins recognize these sites and separate the two DNA strands, creating a structure known as a replication bubble. This unwinding exposes the DNA bases, setting the stage for copying to begin.
key enzymes involved.
Helicase unzips the double helix, separating the strands. Primase lays down short RNA primers, providing a starting point. The main copying work is done by DNA polymerase, which adds new DNA nucleotides. Other enzymes, like ligase and topoisomerase, help join fragments and relieve tension in the DNA.
Copying each strand.
Replication follows a semi-conservative model. Each original strand acts as a template for a new strand. The leading strand is copied continuously, while the lagging strand is made in short segments called Okazaki fragments. These fragments are later joined together to form a complete strand.
Checking for errors.
Proofreading is built into DNA replication. DNA polymerase checks its work as it adds new bases, fixing most mistakes immediately. Additional repair mechanisms catch almost all remaining errors. This high level of accuracy helps prevent mutations and keeps genetic information stable.
Completion and cell division.
Once replication is complete, each new DNA molecule has one old strand and one new strand. The cell now has two identical sets of DNA. This precise copying allows the cell to divide safely, ensuring each daughter cell gets the full genetic blueprint it needs.